Mutations can occur at a lot of different scales. They can occur just at the scale of a single nucleotide changing. They can be at a single amino acid that once coded for one amino acid and now codes for another, or at the scale of a single codon might be a better way to say it. At the single gene or part of a gene can get cut out during a transposon moving around. And also mutations can occur at the chromosomal level. And these often have either very dramatic effects. They prevent an organism from developing, but they're also the engines of creating new copies of genes, of creating new genes, and they're really important for evolution to occur. So chromosomal mutations are large-scale changes to the structure or also the number of chromosomes or to multiple chromosomes. So it could involve one chromosome. It could involve one chromosome giving part of itself to another chromosome. It could involve the whole chromosome being duplicated. It could involve the whole genome being duplicated. We'll mostly focus on what happens when something happens to one chromosome and maybe part of that chromosome ends up somewhere else. So what does this do to our genome when we start having different types of chromosomal mutations? So for example, we can have a translocation, which is say we have a parental and maternal chromosome, and we're going to look at this copy here, and something that can happen is part of this chromosome can break off. So we're going to call this C1 parental. This is going to move over there, and it can actually glom on. It can insert elsewhere. So this results in a deletion on this parental chromosome, and genes are lost, and a duplication, genes are gained on this maternal chromosome. So we lose genes from this chromosome, they're gone totally from that chromosome, and then now there's two copies of each one of these. And often what will happen is when genes get duplicated on a chromosome or within a genome, those new versions of the gene can be mutated through other mutational forces, insertions, deletions, that sort of thing, and then they can take on modified or even new functions. Keyword here, this is large. This is thousands of base pairs, hundreds of thousands of base pairs, whole parts of chromosomes breaking off and moving. That was a translocation. An inversion is when part of a chromosome flips. Again, this is a very large scale, thousands, tens of thousands, hundreds of thousands of bases, and formerly this whole chromosome was oriented 5' to 3'. An inversion happens, and now this section is flipped. And now all the genes that used to go this way now go that way. One thing to think about is, were any genes lost? The answer is no. All the genes that were originally here are still in this modified chromosome. However, crossing over will now be modified. This part of the chromosome will have a hard time crossing over with the original version of the chromosome in diploid organisms. And that change in crossover can impact genetic diversity. So again, what does this do during DNA replication? Nothing. Transcription? Nothing. Rarely can, if these breaks happen in part of a coding sequence, if they happen in a promoter, then maybe two genes at most can be impacted. But crossing over, it prevents crossing over within that region. Translocation is when part of a chromosome moves onto another part. So here we're going to move this arm over there. So previously we'd looked at what happens when two the same chromosomes share parts. This is when, say this is chromosome 1, this is chromosome 2, and then this part of the chromosome ends up on chromosome 1. So whole arms of chromosome, whole parts of chromosomes moving around, moving genes. So often there's some sort of DNA damage, and then DNA repair mechanisms then add the gene on there. So damage occurs to the gene, and then DNA repair mechanisms actually DNA happens to part of the chromosome, the arm floats around inside the cell, the DNA repair mechanisms glue it back on. So chromosome 1 is now longer, chromosome 2 is now shorter. Again, this can impact crossing over and can impact how chromosomes pair up during mitosis and meiosis. At most, maybe one gene might be impacted. The bigger implication is the change in crossing over. So it's important to think for chromosomal mutations, it's important to think for all mutations, what happened to an individual gene? Has its size or length or the number of genes been modified? Has the ability to cross over? Has the ability to cross over been impacted? Has mitosis or meiosis been impacted? And actually, this goes on and impacts speciation, because when you have part of your population that has one form of the chromosome, say without an inversion, and you have another part of the population that has a chromosome with an inversion or with a translocation, then when individuals from those two populations meet, pair up, crossing over is going to be difficult when their offspring later try to produce gametes, and that's going to have implications for speciation that we'll talk about further down the road.